welcome. Today we have with us Dr. Romina Bevacqua. Sorry, I'm a little tricky on the second name. Correct me if I'm wrong. She's an assistant professor at the Diabetes Metabolism Institute, DOMI, and Regenerative Biology and Stem Cell Institute at ICANN School of Medicine at Mount Sinai, New York. And today she's going to talk to us about CRISPR gene editing in primary human islet organoids, and just a little bio of to introduce her. She has a really extensive background in the field. Since January 2023, she's been a new assistant professor in the Department of Medicine, Division of Endocrinology, as I mentioned, DOMI, and the Regenerative Biology and Stem Cell Institute at Mount Sinai. She was selected as a biomedical laureate. Previously, she was an instructor and a postdoctoral fellow in the laboratory of Dr. Sung Kim at Stanford. And her research focuses on understanding the regulatory mechanisms governing pancreatic islet cell function and maturation, and integrates modern genetics, cell and developmental biology, biochemical and physiological approaches. Her, just letting people in. Her postdoctoral research has produced multiple publications, including a first author studies in genes and development in Nature Communications in 2021. The G&D study identified novel functions and targets of two transcription factors. Sorry, there's a bunch of people coming in. SIX2 and SIX3 in human islet B cells. So the Nature Communications study describes the first application of CRISPR-based strategies in primary human islets for site-specific gene targeting, including targeting of presumptive regulatory regions linked to diabetes risk. Her prior doctoral research in Argentina focused on pre-implantation embryo development of large mammals and used modern methods to target genes in embryos. And her studies were externally funded by several agencies, including a U.S. Fulbright Fellowship and by an independent postdoctoral fellowship grant from JDRF. At Mount Sinai, she's investigating how transcriptional regulators, non-coding regulatory elements, epigenetic and external signals orchestrate crucial steps in human islet functional maturation and proliferation, and how these steps are misregulated in human diseases, particularly in type 1, also though in type 2 diabetes. And her goal is to advance and deepen the fundamental understanding of islet regulation and function and their connection to complex traits associated with these diseases. Her recent development of genetic systems, including using CRISPR-Cas9 in primary human islet organoids, termed pseudo-islets, now provides unprecedented methods to investigate mechanisms regulating functions of mature islets. And they have, I'm going to drop the Bevacqua lab link in the chat. They have some really interesting projects that I'm sure many would be interested in checking out. But without further ado, thank you so much for joining us, Romina, and really interested in this talk. This is really sort of opening the door to examine some really elegant methodology.
Thank you, Monica. Thanks, The Sugar Science, for the invitation today, and thanks for that kind introduction. I think you're doing a fantastic job at bringing together the latest research in the type 1 diabetes field and the community, so thanks for that. And today I will be talking, as Monica mentioned, about CRISPR gene editing tools and their application to primary human islet organoids. I think this is a bit of an informal setting, so if anyone has any question, please feel free to interrupt as we go. And just for starters, during the last 15 years or so, we have witnessed really a huge advancement in our ability to both procure and phenotypically profile human tissues. And in the case of islets in particular, this has been made possible thanks to both islet procurement and distribution of islets for research to large number of centers, and also to advancements in the next generation sequencing technologies that now are making it possible to molecular profile islets, not only from healthy individuals, but also from deceased donors, including type 1, type 2, other kinds of diabetic phenotypes, and so on. So these efforts are ongoing, and there are a lot of groups and a lot of consortium working together to profile these islets, to identify their transcriptomic profile, their epigenomic profiles, proteomics profile as well, and the genetics and all the functional mechanisms that might be affected and not properly working in these deceased donors. So putting all this data together, it has been possible to start identifying which are the pathways, which are the gene regulatory networks, which are actually not working properly in the deceased donors. And this is also calling for a need for systems that now allow us to actually study these novel mechanisms in the human islet context and see if with these studies we can actually move things forward in a more fast fashion and be able to identify novel therapeutic targets and improve our efforts with the stem cell-derived beta cells, for example. Of course, we have models. We have mice islet systems, and there is a lot that has been done and a lot that we have learned using the mice system, but there are many differences between human and mice islets. There are differences in their architecture and development, and this can be exemplified here just by showing and observing this mice islet, which, as you can see, has the islet insulin positive cells in the core surrounded by the alpha cells, whereas in the human islet, the different endocrine cells are intermingled. And more recently, by means of profiling the transcriptomic and proteomic profiles of human islets, we have also identified transcription factors, for example, such as SIX3, which I studied during my postdoctoral studies, which are highly expressed in the beta cells of human islets and which are required for the proper function of these adult islets, which are not expressed in the beta cells of mice, for example. So all of this calls for a need to actually adapt systems to the gold standard in human islet biology, which is the study of primary human islets. And with this purpose, we have generated this human pseudo-islet platform, which basically consists of the dispersion of human islets and allowing these dispersed islet cells to reaggregate spontaneously into what we call pseudo-islets. So we can perform transduction with lentiviruses of these dispersed islet cells, coding for example, short hairpin RNAs and reporter genes. And in this way, we can knock down the function of genes of interest. And we can also adapt this system to overexpress genes or cDNAs of interest. So only 5 days after the specific transduction with our lentiviruses of interest, we obtain these human pseudo-islets, which, as you can see here, are very similar to the primary islet counterparts, albeit a bit smaller in size, and they express the reporter gene at high levels. Only 5 days after the transduction, we can evaluate in vitro the functional consequences of the knockdown or overexpression of our gene of interest. And we can perform, for example, functional studies such as perfusion or a static batch assay. We can perform transcriptomic studies. We can do sorting. There are a bunch of things we can profile only 5 days after the generation of our pseudo-islets. But we can also transplant under the kidney capsule of immunocompromised mice and study in this way the longer-term consequences of this knockdown by means of IPGTT or intraperitoneal glucose tolerance test 30 days after transplantation. We can also recover these grafts and study these grafts by immunohistochemistry or even sequencing studies. And despite the many advantages of this system, there are some sort of questions that we cannot study using knockdown or overexpression. And this is mostly linked to when we are interested in understanding genetic mechanisms. So when we want to study the genetic predisposition to diabetes and study variants that are linked to an increased risk of diabetes, or when we want to model monogenic forms of diabetes, we actually need to target the DNA. And that's how we became interested in adapting CRISPR-Cas9 technology for a specific gene targeting in the primary islet. For those who are not very familiar with this technology, I wanted to provide a bit of background. So the CRISPR-Cas9 system actually, and the CRISPR-Cas systems were actually discovered in bacteria and archaea, and are part of the adaptive immune system of this bacteria and archaea. And it's a way in which these cells destroy invading or exogenous viral DNA or other foreign DNA. And the main discovery that made it possible, the adaptation of this immune system into a gene editing tool, was the finding of the type 2 CRISPR-Cas system, which is the CRISPR-Cas9 system, which is based on a single nuclease, which is Cas9, that can actually target any genomic sequence of choice. And then, thanks to combined efforts and knowledge by Jennifer Doudna and Emmanuelle Charpentier, which granted both of them the Nobel Prize in Chemistry in 2020, they were able to actually generate this single guide RNA, which with only one single molecule of this guide RNA, we can target this Cas9 protein to virtually any genomic sequence of choice. And the only requirement that the system has is the presence of a PAM sequence in the form of NGG that Cas9 needs to recognize 3' from the single guide RNA sequence that it's targeting the genomic DNA. So after Cas9 is brought to this specific gene location of choice, it will induce a break in the DNA, and then it's the endogenous DNA repair machinery which is going to repair this break. And by doing so, and the more traditional way in which the cell will repair a double-stranded break is using the non-homologous end joining approach, it will introduce small insertions and deletions, so the repair won't be actually perfect, and this will generate a shift in the open reading frame of the protein, and in that way we will either introduce an earlier stop codon or will truncate the protein, and so the protein function will be lost. And using this kind of approach, it has been shown that many different cell lines can be targeted, different model organisms. Here we are showing zebrafish, but it's zebrafish, mice, livestock, props, bacteria cells can be targeted. And this has really made a revolution in gene editing approaches. But as it usually happens, when we want to adapt this technology to primary tissue, to non-dividing cells, things become way more tricky. And we were only able to report the first work applying this technology to primary islets in 2021. And so today I'm going to start by talking about our initial work with CRISPR-Cas9 based on lentivirus, and then I will briefly touch base in what we did more recently, adapting this technology with a Cas9 ribonucleoprotein complex. So as a proof of concept, the first target that we chose was PDX1. So we designed single guide RNA, which was specific for PDX1, and we evaluated whether the CRISPR-Cas9 system could actually induce a knockout in this specific genomic location. Why we chose PDX1? Well, it's the first transcript to be expressed in the developing pancreas. In the adulthood, it becomes restricted to beta cells, and heterozygous mutations result in a type of diabetes known as MODY4 as a result of haploinsufficiency. So we thought it was an easy initial candidate to try gene targeting. So I think the step in which we spent more time was to see how to deliver these elements of the CRISPR-Cas9 system. And ultimately, we chose this lentiviral-based approach composed of all-in-one components of the CRISPR-Cas system. So it codes for the single guide RNA under U6 promoter Cas9 under a CMV promoter and GFP for selection. Using this system and transducing our dispersed islet cells with these lentiviruses, we were able to generate human pseudo-islets. And of course, the first thing we did was specifically sort the GFP positive cells for further analysis. So question number one, can we actually achieve efficient targeting of PDX1? So we performed a PCR with primers surrounding the genomic sequence of the single guide RNA, and we sent this for sequencing. And as you can see here, underneath this red line is the single guide RNA sequence. This is the PAM sequence I mentioned before is necessary for Cas9 to induce or to recognize and then induce the double-stranded break, and usually 3 base pairs upstream, Cas9 will induce the cutting in the DNA. If we look at the bases here, it's exactly in that position where we start to see double peaks, indicating that Cas9 indeed induced the cut and it was repaired, and this is altering the sequencing, the nucleotide sequencing. So we calculated the percentage of indel-modified sequences, and we observed that over 60% of our sequences were actually modified, suggesting that, yes, indeed, we had efficiently achieved targeting of PDX1 using CRISPR-Cas9. But of course, we wanted to verify that the protein was being reduced, and we did that by comparing bioimmunohistochemistry CRISPR controls versus CRISPR PDX1 target pseudo-islets. And as you can see here, there is a reduction in the levels of the PDX1 protein. And this also correlated to impaired islet function in vitro. So we tested the expression of transcripts that we knew are downstream of PDX1 based on studies from mice, and they were all downregulated. And we also performed functional studies. So when we did these functional studies in vitro, and this is an example of perfusion, we observed that PDX1 knockout pseudo-islets had lower secretion of insulin compared to the control counterparts. But this was even more striking in vivo. So when we transplanted these pseudo-islets under the kidney capsule of mice and we performed IPGTT studies 30 days later, we observed a significant decrease in the serum human insulin in these mice. So altogether, our results so far were highlighting that CRISPR-Cas9 can be applied to our primary human islets, and we can actually apply it to non-dividing cells. But we were more interested in adapting this tool not to target a transcription factor necessarily or a protein coding gene, but also to study non-coding regulatory regions linked to diabetes risk. And in this case, we were interested particularly in a presumptive regulatory region in the 6.2/6.3 loci. Why we chose this regulatory region in particular? Well, as I mentioned before, 6.3 and the same happens for 6.2 are not expressed in the beta cells of mice. But in addition to that, both of these transcription factors show differential expression between juvenile and adult beta cell stages. What's interesting is that if we overexpress 6.3 in beta cells from juvenile islets, it results in enhanced insulin secretion. But when we knock down the expression of these transcription factors in the adult beta cells, we impair the islet function. And actually, before knowing all this, what we knew was that there were variants in the 6.2/6.3 loci that were linked to fasting hyperglycemia and increased risk of type 2 diabetes. So really understanding which are the regulators of the expression of these transcription factors that only start to be expressed later in life is relevant for beta cell adult function. So
Romina, can I just ask you to qualify for the juvenile group? What age group is that?
These are islets of less than 10 years of age.
Great, thank you.
So it's postnatal, yeah. And so when we went on into looking at where these risk SNPs were falling, it was interesting to see that they were actually overlapping a presumptive regulatory element, which we are calling, starting from now on, as 6E. And what we didn't know for sure was whether actually this regulatory element is regulating the expression of 6.2 and 6.3, or which is its function. Considering that we don't see expression of these transcription factors in mice, we cannot study this in mice, and considering that the expression of them is also only in adult stages, we cannot actually use stem cell models or cell line models of human beta cells to study this mechanism. So really having the tools of CRISPR adapted to primary human islets is key to study this kind of mechanisms. And so we adapted and the CRISPR-Cas9 approach I showed before, but for the insertion of deletions. So we designed 2 single-guide RNAs for CRISPR knockout. And as I'm going to show later, we also adapted the tools for CRISPR-A, which is CRISPR activation, but that's in a couple slides. So for the introduction of a deletion of this regulatory element, we adapted the construct to express 2 single-guide RNAs instead of only 1, and the rest of the construct remained the same. We evaluated the expression of the presumptive targets of this non-coding regulatory element, and as you can see here, both 6.3, the 6.3 antisense, and 6.2 showed reduced expression, whereas other neighboring genes of this genomic region were not changed in their expression, showing that this regulatory element specifically targets these presumed target genes. In order to further understand whether this was the mechanism and also to see this from a complementary approach, we adopted another approach possible with the CRISPR-Cas9 system, and this is CRISPR activation. So what's the difference here? That Cas9 does no longer have a nuclease activity. So this is a mutated Cas9 where the nuclease activity is mutated, so it can no longer introduce a break in the DNA, but instead it's fused to a transactivation domain, which is highlighted here by BPR and BP64. So these are activation domains that are fused to this Cas9. So by binding to the specific genomic location of choice guided by the single guide RNA, instead of cutting the DNA, it will activate the expression of this region. And it can be either a promoter region or an enhancer region. So we use this, again, all-in-one lenti-CRISPR system with 2 single-guide RNAs because CRISPR-A has shown to work more efficiently when you have 2 or 3 guides targeting the region of choice, and the dCas9 and GFP for selection. And using this, we target the enhancer, and what we wanted to understand was whether our presumed target genes, 6.2 and 6.3, were actually showing an increase in their expression and what was happening with neighboring genes. And we saw exactly what we were hypothesizing and what our other findings with the deletion were showing. So 6.2 and 6.3 were showing an increase in their expression, and other neighboring gene was actually not changing the expression. So altogether showing that we can adapt this technology also to target non-coding elements and to functionally validate the role of these presumptive regulatory regions linked to diabetes susceptibility. So in summary, so far, we have shown that we can target pancreatic islet cells with CRISPR-Cas9 in vitro, both to induce indels, deletions, gene activation. We can target both coding and non-coding regions. And what remains is to try to adapt this to a more simplified system in which perhaps we don't require anymore to generate a lentiviral construct for each target we want to apply. So that's what led us to this new work, trying to adapt CRISPR-Cas9 ribonucleoprotein electroporation to the human islets. Which are the advantages? As I mentioned before, simplicity. We just order these single guide RNAs. We don't need to clone them. We don't need to make lentiviruses. It's more easy to scale. We don't need a BSL2 plus setting in the lab. We are also avoiding the lentiviral integration because we are not, with lentiviruses in a random position in the genome, we are integrating these lentiviruses. Using this system, we are not leaving any footprint after the edition other than the specific gene targeting of choice. And this has a lot of potential applications. And the other advantage is that we might be able to adapt this technology for multiplex targeting and the targeting of more than one location simultaneously. So I will very briefly show what we initially did to see if we could apply the electroporation approach. And then I will very briefly touch base on our multiplex approach on a PCSK1 regulatory regions. So how does this work? So we form in vitro a complex between Cas9, which can be fused to GFP or not. We initially used this one just to see something. On day one, you can see the formation of the complex and the efficient electroporation into your cells. So after formation of the complex, we mix them with the dispersed islet cells. We electroporate them, and for this purpose, we use the neon system. And then we again allow them to reaggregate into the human pseudo-islets. One thing here is that we don't have a reporter gene that will be expressed until day 5, so we cannot sort. But I think that's also one interesting finding here, that the efficiency is kind of so high that we still can see the phenotype without sorting of the efficiently electroporated cells. So we use the same single guide RNA as before, and so after translocation into the nuclease, Cas9 induces a cut in the human PDX1 exon 1. We measured the efficiency of the cutting, and in this case, the efficiency was in the order of 40%. So we decided to pursue further with this approach and study the possibility to target regulatory regions. And we chose PCSK1, which is which codes for PC1-3, which is responsible for the conversion of proinsulin into mature insulin in human. And what is interesting is that the transcription of PCSK1 is regulated by glucose. So we could potentially study a mechanism regulated by glucose after targeting with our CRISPR components. What's also interesting is that the regulatory regions of PCSK1, both the promoter region and an enhancer that has been linked to PCSK1, are also showing increased H3K27 acetylation at high glucose concentrations. So it's likely that these two regulatory elements are the ones that have higher accessibility and thus regulate PCSK1 for it to have a higher expression at high glucose levels. So we designed single guide RNAs to target either the promoter or the enhancer. And of course, the first thing we did was to evaluate the efficiency of these targetings. And again, we are not sorting for cells here. We are just evaluating the entire population. So we performed a PCR of the region flanking the target sequence by the two single guide RNAs on the PCSK1 promoter region, or the target region in the PCSK1 enhancer region. In both cases, the percentages of sequences showing the specific modification were in the order of over 85%, as you can see here. And while when we target the promoter, the efficiency of the targeting of the promoter was in the order of 85%, the enhancer was not modified as expected, and inversely occurred when we target the enhancer. So the enhancer sequences were modified, but not the promoter ones. What happened when we target both? Well, both sequences were modified with high efficiency, and that's what you can see here in the green bars. So altogether showing that not only we can achieve efficient targeting using the CRISPR ribonucleoprotein approach, but we can also perform multiplex targeting. And then we wanted to study how the targeting of these regulatory elements affects PCSK1 expression regulated by glucose. So after inducing the targeting, we cultured our islets at either low or high glucose concentration and evaluated the levels of expression of PCSK1. And as you can see here, after targeting of PCSK1 promoter or PCSK1 enhancer, we didn't see changes in the relative expression of PCSK1 at low glucose concentration, but we observed a significant decrease in the expression of PCSK1 at high glucose concentrations when we target either promoter or enhancer. But when we target both together, enhancer and promoter, both at low and at high glucose concentrations, the expression of PCSK1 was impaired. And this also, we evaluated phenotypes after this, and we observed that proinsulin was increased in all these conditions. So in summary, CRISPR-Cas9 using both lentiviral and electroporation-based approaches can be used for a specific gene targeting of adult human islets. We can study coding DNA, we can study non-coding elements, synergistic effects of distinct regulatory elements. We can introduce indels, deletions, induce gene activation, and we are confident that we can introduce different kinds of modifications that we will be starting to explore soon. So we are expanding really our genetic toolkits for adult human islets. And in my lab in particular, we are interested in adapting these tools towards the study of regulatory mechanisms that govern the adult human islet function. And I think these are complementary studies, very necessary to better understand what makes an adult beta cell an adult beta cell and what is dysregulated in disease. So if you're interested, please check my lab website, and we have positions available. With that, I would like to thank the members of my lab, the members of my prior lab at Stanford, and our collaborators, including the collaborators now here at DOMI. So with that, thank you very much. Thanks to the donors and their families for making this possible, and for the islet distribution agencies.
Wow, this is a really great new protocol that you guys have sort of unearthed and brought to bear. I think, I mean, you can see that it's going to have so much use by many different interdisciplinary scientists, and you can see that it's really quite adaptable to different lines of study.
I hope so. I think that's our goal, that we can actually use these tools to better understand the functions that are dysregulated in disease, and hopefully help and lead the way for the identification of novel druggable targets.
Yeah, I mean, if we talk about the studies that you have, the projects you have currently, you have 4 major projects listed on your website. The project 1 is identification of transcription factors and pathways driving pancreatic islet maturation, which you kind of detailed using, the first steps using this tool. Project 2, deciphering non-coding regulatory mechanism driving islet cell maturation and dysregulation, again, right, applicable. Project 3, elucidating external signals regulating pancreatic islet maturation. Again, the tool could be probably used. Project 4, development of novel in vitro and in vivo genomic approaches to reverse diabetes. So the tool, seems like the tool really could be, used across all those project lines.
Yeah.
And, specifically what I find, so powerful is that you really are you really disseminated the differences between mice and humans, and the fact that this is specifically targeted at islets that are derived from humans. So it's amazing. Here is a question that just came out. What have been some challenges using electroporation? And I actually had a question around that as well. Like, did you have to really play with the electroporation to get the sweet spot?
Yes, yes, definitely. So we tested a bunch of voltages and concentrations of the ribonucleoprotein single guide complex. If you go a bit too low in the complex, you don't actually achieve efficient target. If you go a bit too high with the voltage, you kill all the cells. So it's, yeah, that's one of the things to fine tune. And we started or to know where, like to set the initial step, I looked into other primary cell models. So I looked into some published work on T cells, which they are way different, but at least they are primary, they are non-dividing, and I used their conditions as a start point, but even so, we had to change them quite a bit to find the ones that were better for us.
Yeah. And I'm sure that that protocol is detailed in the paper, correct?
Yes, yes, yes.
Great. And I wondered, with you have a graduate student and postdoc. You have a senior research.
Emilio is he's a senior postdoc and a senior research scientist in the lab. Yes, and he's now doing all this. So he's doing all the electroporation approaches, and also we keep working with lentiviruses, depending on the kind of approach we are doing, what we chose. So Emilio is there. You can say hi.
Hi, Emilio. It's great to know that there's, a senior research associate in the laboratories along with yourself. And I noted, you noted that you're looking for postdocs and graduate students. So if you'd like to get that out to our global, audience, please feel free. We have a mechanism on our website for doing so, where you can contact us directly. Well, thank you so much for this. This was a really, really interesting talk, and it really inspires me to sort of dive back into start thinking about all the different regulatory elements that could be potentially targeted with this, and just sort of thinking through there's so much work there. So
yeah, really.
but it would be really, really, it's really a powerful approach. So thanks so much for bringing it to bear. And you have a lot of really great accolades in the chat here. Incredible job, amazing. Thank you so much. Interesting approach. So I think I'm not the only one appreciative of this approach. I hope you have a great rest of the day and a great holiday. And we'll be looking for the next series of papers that come from your lab.
Thank you. Hope so. Not soon, right?
Well,
we
yeah,
but
soon enough. Okay,
yeah.
bye-bye now. Have a great rest of the
Bye.
day.
Thank you very much and happy holidays.
You too. Bye-bye.